Well, in the last previous two session, we discussed about the replication of viruses, and all the different stages of virus replication, and then I requested all of you, like, if you have any doubt or query, you can put some questions. So I got one question here where it is asked, like, the endocytic pathway and how the pH effect on it. So this particular process we discussed when we talk about the penetration and uncoating of viruses. So the different process of endocytosis means the crossing the membrane, the virus when they get enclosed into an endosome inside the cytoplasm by different process like pinocytosis or by caveolar or raft mechanisms or by clathrin coated pits. The virus particles were internalized, or they cross the membranes and they come to the cytoplasm within an endosome. Okay, so not all viruses. There are viruses where they do not enter through this endocytosis process. Rather, the virus envelope get fused with the plasma membrane by the process of fusion, and then the nucleocapsids, they enter into the cytosol. Okay, so certain viruses, they follow this endocytic pathway. So starting from the cytoplasmic membrane, that they get endocytosed within the endosomes, cytoplasmic vacuoles, and then near to the nucleus, say. And in this process, when these endosomes, they remain near to the cytoplasmic membrane, you can see there the pH is almost near normal pH of 7.4. It is showing through this color strip here. And gradually when they migrate, then because of the activation of certain process like M2 ion, our discussion, through which generally what we can say is that within these endosomes, some the hydrogen ion concentration increases, and that leads to a decrease in the pH level. So gradually towards the nucleus, you can see this this endosome sometimes diffuses to the lysosome if present inside the host cells, or otherwise even in the late endosomal phase, it become highly acidic, and that triggers the disintegrations of the capsids, nucleocapsid structure of the virus, and the virus nucleic acid get released. And that is the process what we call as that uncoating of viruses, and this process we call as the endocytic pathway. Of course, I'm just telling it in a very simple way. However, several factors plays a role here. In some case of the endoplasmic vehicles also takes place that leads to acidification of this endosomes. Otherwise, like the lysosomal granules, if present inside those cells, then they can be fused to form these endolysosomes, and that triggers the uncoating process. Okay. So here it is another picture. So it is showing like just near to from the cytoplasmic membrane, these endosomes, you can see the range of the pH. Gradually it falls down as it migrates deeper inside the cells. Okay, so this is a process what we call as endosomal maturation, and finally this maturations give rise to breakdown of the nucleocapsid structure. Okay, basically the fall of pH will trigger the capsid protein binding loosens, and the capsids get dissolved. The capsomers get disintegrated, and the nucleus here it is another examples of say adenovirus. These again pH dependent process for encapsulation. So through this pinocytosis, it will just come to the cytoplasm, and as it might to deeper, then you can see that there is fall of pH inside these endosomes. Some chain reactions gets triggers, and that leads to influx, and that leads to fall of pH, and finally that will cause breakdown of this nucleocapsid, and the nucleic acid will be released. So finally the nucleic acids will be released to the cytoplasmic mass. So in case of the DNA viruses, as I said, that encapsulation will take place just near the nuclei, and then they will migrate to the nucleus to initiate its replication. Right? So the list of certain viruses takes place, and the viruses, they simply penetrate through the cytoplasmic membrane and come to the cytoplasm, the cytosols. So they don't depend on this fall of pH for encapsulation. So in the uncoating, there's several phases that happen. So first thing is that depending on the type of virus where it will replicate, it will take place in a correct location within the cells. So the conformational changes, more particularly proteins, will lead to progressive of the capsid proteins and loosening of disulfide bonds will cause disintegration of the capsid layer. Okay, so nucleic acid will get released. So this is a classical example of influenza virus. This diagram I'm showing it here. So when it enters through this cytoplasmic membrane and through these early endosomes, and gradually the pH fall down will takes place, and this is basically because of activation of one phenomenon where the hydrogen ions, they will influx, as well as the potassium influx takes place, that triggers the fall of pH, and gradually this the hemagglutinin, they will get fused with the endosomes, and just near to the nucleus they will get released. Okay. Hope that this information is okay for you. Then there was another question. Here say that if the DNA virus replicates in the nucleus, then why protein synthesis occur in the cytoplasm? It's not like that. See, protein synthesis always takes place in the cytoplasm. All the protein synthetic machinery like the ribosomes and other protein synthesis factors are present in the cytoplasm of the infected host cell. So this is an example of a virus like DNA virus, HepaDNA virus. So when it enters and finally the nucleic acid gets released inside the nucleus, then the transcriptions will take place. The messenger RNA will be produced like early messenger RNA, and then this messenger RNA will come to the cytoplasms where on the surface of the ribosome, the protein synthesis takes place. You know, the protein synthesis occurs in the cytoplasm, not inside the nucleus. That's the reason, okay? Probably you're confused with this particular diagrams where I have shown it that this is again the type of virus, the herpes virus. There's, they first early proteins will again come to the nucleus, and where the genomic replication will takes place. Then the late messenger RNA, late proteins, and these late proteins are again migrating here for assembly, because this virus, the entire genomic replication, active space, as well as the assembly that takes place inside the nucleus. But it's not so with all DNA viruses. For example, this is another DNA virus where the nucleic acids goes inside the nucleus, the transcriptions of a messenger RNA will takes place, they will come to the cytoplasms where the protein synthesis will takes place. Then these proteins will not begin migrates because their factors are available in the cytoplasm, and they will go on assembled here, and finally through this endoplasmic reticulum pathway they will comes out of the host cells by the budding process, and then blood viruses will be released. Okay, so I think this will satisfy your queries and doubt. May I know who has texted me? Your name was not. Hello. Well, Brookesh. Hello. Anyone, please respond.
Yes, sir.
Yes, sir.
Could you see the slides and could you?
Yes, sir.
Hello.
Hello. Yes, sir.
Yes. Can you see the slide?
Yes.
Yeah, this question text me yesterday. Me and could you be able to understand the doubts? Okay, so please and do interact and put some queries if it is not understandable to you. So if it is so, then we can shift it to the next topic as per our syllabi. Yes. Yes.
Could you let other like there are students who want to enter the classroom?
Yes, I understand. Okay, give me that. Well, today only 40 of your friends joined the class. So it's like the virus cell interactions. When a virus infection takes place inside the host cells, if you understand like when they infect a particular cells inside the host, and what are the things that may happen into that cell. So it's not that all the time, but the viruses infect a cell, the cells will undergo disintegration and they will die. It may not happen so. So depending on the type of viruses, we could see several different type of virus cell interaction. So, you know, like the viruses, when they infect a particular host cell, always they first try to shut down the host cell functions. The reason so is that they need the host cell machinery for their own synthesis. So always they are in a tendency, there is a tendency to shut down the host cell function. But again, the host cells will die before the virus could able to replicate. So there is some way in the processes where certain viruses, they shut down this process moderately. Some of the viruses, they go to a higher extent. So likewise, the viruses involved in the...
Excuse me, sir.
I could not see anyone here. Okay, two more. Okay. So by understanding these virus cell interactions, what are the different consequences? If we know for a particular virus, that even help us in understanding the disease produced by that particular viruses into in a species of animal. Okay, so whenever there is any virus enter into the host cell, definitely the host defense mechanisms will act to eliminate the infection, right? So in that process, again, there are a lot of changes that occur. Most of the time, the virus infections may remain undetectable, means that the immune systems will be can eliminate the virus infections and no any clinical manifestation may occur. So undetectable infections may occur in certain virus cell interaction. However, some virus infections, we can see that the host defense mechanism may not be able to totally control that growth and replications and pathogenesis of the viruses. So some clinical manifestation, a course of disease may occur, and afterwards the persons may recover from infection. However, there are certain infections where it is lethal, means the virus infection or the viral cell interactions may lead to the death of the individual. So the understanding about these different aspects of viral cell interaction is important. So collectively, we can say that the virus and the cell interaction, with all the different viruses that causing the diseases in man and animal, we can say that the interactions may be cytocidal interactions. Cytocidal means the virus infections will lead to death of the cell by different mechanisms. The virus can induce death of the cell. It's called as cytocidal. The virus infections will directly cause death of the cell. There are several mechanisms through which they can induce the death. However, there are many virus infections where they do not cause death of the cells. We call them as a non-cytocidal interaction. So cytocidal and non-cytocidal interactions. So in the non-cytocidal interactions, again, we can see there are certain once the virus infection and the acute phase of the disease is over, the virus may remain inside the host for a prolonged period. So, and gradually the virus particle will be released through some excretion and secretions. We call it as a persistent productive infection. Generally, it is a non-cytocidal, means the active cell death doesn't occur, but the virus persists inside the host. They interact with the host immune systems, and the immune system do not or restrict the virus infections to form into the acute stage. So we call this particular type of interaction is the persistent productive. The other is the persistent non-productive. There are certain virus infections where the virus, after getting entered to the host cells or after acute phase of infection, they will persist inside the host without any active virus replications, or they undergo a stage of what we call as the latency. They will simply hide inside the host and no active virus particle will be produced. This is also a non-cytocidal interaction. So what we can say that the virus cell interactions is maybe cytocidal, where the virus infections leads to death of the cell, infected cell, or there are certain infections where inside the host, so the virus will persist for a prolonged period after the acute phase of infection. So we call it as a persistent productive, and slowly the virus particle will be released from that host to the environment. The third one is again a kind of persistent infection. Persistent means after entry of the virus will replicate, and after that the virus will persist within the host for a long period, and no active virus particle will be produced. Is called as the persistent non-productive, or sometimes we call it as a latent virus infection. So this is very crucial while we talk about the elimination of virus infections from the population. So we could see certain viruses where after entry into the host cells, they can simply undergo a stage of latency, or they can persist without the production of active virus particles for a prolonged period. So this kind of viruses creates problems, and the animals who are persistently infected become a source of infections for other animals. As a result, the disease persists in the population. Okay, so these persistent productive and non-productive, these are non-cytocidal virus cell interaction. Okay, where basically the no architectural or cellular changes that takes place. Those infected cells may lead to some of the physiological changes in the cells, but the cells will not die. Okay. Then the last type of so this referred to uncontrolled proliferation of a cell, or in simple language we can say it is called as the tumor formation. So there are certain identified virus which both in the DNA virus and RNA virus category we are going to learn in the subsequent classes, which when they infect a host cell, then they can induce tumor or uncontrolled proliferation of the cells. The cell transformation takes place. Okay, so it has now been established that there are certain viruses which can induce cancer, this uncontrolled proliferations that leads to tumor formation in animal and human. Transformation. So basically, all the different type of virus cell interactions we can categorize for our easy understanding as cytocidal, persistent productive, persistent non-productive, and transformation. Now, with a few examples that's been given it here, of course, we are going to discuss about all these viruses in our subsequent classes. So in the cytocidal, basically what happened, this the cell morphology and architectural details will lost. So there will be a time where the virus, presence of the virus, or the growth of the virus inside the cells will so badly affect the host cell. So they cannot sustain this injury, and finally they will die. And the active virus particle will be produced, and they will be infecting other host cells. So likewise, the cellular damage can be initiated by presence of the virus. So we call it a cytocidal virus cell interaction. The second category, as we discussed about the persistent productive. So here what happened in the persistent type of infection, the virus doesn't cause any but the cells continues to divide the host cell, but sometimes they lost some special function. But it takes place, then secretion, granular secretions may happen, a production of hormones may get affected. But the cells normally they continue to divide. The virus also persists within that cell. So this persistent productive infections, the infected cell will gradually secrete or produce some mature virus particle, which will be released to the surrounding. And so there, in the persistent productive, some of the specialized function, particularly about the disease like rabies. So the neuronal damage may occur because of the presence of the virus and the growth of the virus inside this host cell. So nerve impulse and will be affected, and the cells will no more be able to function as a nerve cells. However, you cannot see any cellular architectural changes. So this is what we call as the persistent productive type of infection. Then coming to another category, it's called the persistent non-productive. There are certain viruses, initially they may produce some cytocidal effect, but at the later part of the disease, they may undergo a stage of so in the non-productive stage, no active virus particle will be produced. So the viruses will remain silently, or what we call as the latently. Latently, they will present inside certain species. They did not cause production of new virus particles, As a result, no cellular changes occur. So we call it as a persistent non-productive infection. So some examples are given with here, like the canine distemper virus. After the acute phase of the disease, they can undergo the persistent non-productive stage in the nerve cells. Then the third, the fourth category of interaction is the transformation. As I said, that the entry of the virus and the growth of the virus inside the host cells will lead to alteration of cellular morphology and uncontrolled proliferation of cells. That overgrowth of cells may take place, and that leads to development of visible tumors. And we designate them as the oncogenic viruses. So different kind of oncogenic viruses, they can cause transformation of cell, right? So in earlier classes, many times I repeated the retroviruses, why they are very, very unique in its sense that the retrovirus once infect the host cells, so the viral RNA will be converted into with the host cell chromosome, and that particular stage is called as the provirus stage. Now, insertion of a retrovirus within the chromosomes of an animal may lead to mutation, and then the cells may undergo uncontrolled proliferation, so tumor formation may take place. So we'll have a class on the viral tumorigenesis. There we'll discuss more about this process, how it may occur. The tumor formation may occur because of certain virus infection. Now, if you see a little bit more, if you can get information about the cytopactic or the cytocidal interactions, how a virus can causes damage to the cell. So, we know that the inhibition of host cell nucleic acid synthesis, the presence of the viral proteins, and presence or the replication of the virus within the host cells may severely affect the nucleic acid synthesis inside the host cell. Example, we can talk about the pox virus. They carry, like it's a very large virus having almost 200 different protein they carry. So one such protein is called as the DNase. Then proteins can cause damage, cellular DNA damage to the host cell. Besides the other viruses, they also can inhibit the RNA transcription, cellular RNA transcriptions within the host cell. The host cell RNA transcription own synthesis. Either they may utilize the host cell transcription factor, or some of the virus factors may shut down the transcription mechanism of the host cell. So if they goes up to a level that the cells cannot survive, then we can see the cytocidal changes. Then inhibition of processing of host cell messenger RNA. So the messenger RNA formations is hampered because of presence of certain viruses like influenza, the herpes viruses, and then that particular cells are unable to survive, and they gradually undergo lysis and death. Then even the virus, they compete with the host cell protein synthesis mechanism. So they overpower the host cell protein synthesis process. As a result, they utilize the entire ribosomal surfaces and the transcription factors, and then the translation factors of the host cell protein synthesis get affected, and they will die. So even there are certain viruses where the capsid protein or some of the early proteins become toxic to the cellular compartments or inside the cells. They may cause damage to the actin fibers, and the presence of these viral proteins become toxic, and they undergo cytocidal changes. It may also affect the or the mechanism of damage also attach and causes the membrane damage severely, and more particularly which can cause fusions of the plasma membrane of adjacent cell. As a result, you can see here, like this is an cells and the virus particle when infected express these fusion proteins, the viral fusion protein on the surface of the host cell, and because of which the adjacent host cells will fuse together, and that will give rise to a structures what in virology language we call it as a syncytium formation. So it is a cytotoxic effect. There are many viruses which can produce syncytium. So once this kind of fusions will take place, the host cells will unable to survive for long period and they will gradually die. This is a cytocytal changes that occur through the fusion glycoprotein. So exactly happened with the SARS-CoV-2 virus. This virus is also expressed this spike protein on its surface, and that will again initiate the syncytia formation in the alveolar cells, and cellular damage it may initiate. So there are some of the mechanisms. So going ahead, we could see certain viruses which have the property of, particularly the influenza viruses, they possesses some spike glycoprotein on its envelope, which can bind with the RBCs. So RBCs possess the receptor for those, and the RBC can aggregate on the infected host cell. And this is utilized for even diagnostic purpose of certain viral infections, where we can perform the hemagglutination test or heme adsorption test, more particularly for African swine fever virus in diagnosis. The heme adsorption test is performed, where this kind of satellite formation takes place in the infected cell. The RBC will bind. Okay, so they drastically affect the cellular functions, and the cell death may occur. Then another mechanism through which the cytolysis may take place is by immunological mechanisms. Like certain viruses, they stimulate the host immune system continuously and triggered a very high level of immune reactions. What these days all of you have heard about the cytokine storm produced by this SARS-2 virus, and this excessive amount of the cytokines, when they are triggered by the presence of virus inside the host cells, may lead to the pathological consequences of cytolysis. Although we know that the immunologic response is good for elimination of virus infection, but if it is an uncontrolled stimulation of the immune system, a continuous and high level of inflammatory cytokines will be produced, and they can cause cytolysis. So the virus cell interactions, even the cell death may occur because of immunologic mechanism. This is one aspect. Then the direct changes, like the growth of the viruses can directly, like the polio viruses, the straightway affect and damages the actin fibers, the microfilament within the host cell. So as a result, the host cell, it loses its shape, particular shape. So, and the ballooning of the cells and the fibroblastic or the elongated cells become round. These are some of the changes which can be visible because of the virus infections. Okay, so the microtubule or the tubular microfilament structures, which gives the definite shape and organization of cells, may be disintegrated because of presence of certain virus proteins. there are many viruses which produces some protease enzymes needed for their self cleavage of their own proteins, so that the nucleocapsid structure they can form. These enzymes may have direct effect on the cellular microfilament. So these are some aspects through which the cytopathic changes may occur. Okay. So again, if you just recollect briefly here, like how the cytosol changes may take place. So it's maybe because of inhibition of host cell nucleic acid synthesis, host cell RNA transcriptions effect or inhibition of the host cell RNA transcription, or the inhibition of processing of host cell messenger RNA, host cell protein synthesis inhibition. Then some of the toxic viral proteins may damage the cell. Then the damage the cell membrane that leads to the cell lysis. Then the fusion proteins presence on the surface of certain viruses may causes the syncytia formation, the fusion of two and large type of cells with multinucleated cells, and we call it the syncytia formation, and gradually they disintegrate and they die, the cell. So the phenomenon of hemabsorption and hemagglutination induced by the virus, the cellular damage may occur. The immune mechanisms or immunologic stimulation of the immune systems, which may causes damage to the cells, which can cause death of the host cell or the virus infected cells. Even you have already learned about the activation of lymphocyte causes death or disintegration of infected virus infected cells. So they can induce the all infected cells, they can undergo cytotoxic changes. So by different immunologic mechanism, the cell death may occur. Then again, this growth of the virus within the host cells, the assembly and accumulation may directly affect the architectural changes in terms of the microfilament, the intermediate structures in the host cells, and the cell lysis will take place, which the cytosol changes occur. Now, the non-cytosol changes in the virus-infected cells, we could see that there is practically no damage, or physically we can see no damage to the cells. The cells remain normal, and they are able to replicate and grow and replicate. Even the presence of the virus doesn't affect their growth and replication. But however, the presence of the viruses, either in the productive as well as in the non-productive aspect, may affect the specialized function of the cells. And as I said, that the nerve cells infected with certain neurotropic viruses will lose their capability to transmit the nervine pulses, and they may lead to a condition called as paralysis, where it is not because of the cell death, it is because of specialized functions get lost because of the presence of the viruses inside the host cell. Right? Then, the virus-induced cell death. This is an innate mechanism. Whenever the virus infections takes place in any body cells, they will undergo the apoptosis or programmed cell death. This is a defense mechanism by which the cells try to disintegrate in a programmed manner, and so that the virus cannot able to complete its life cycles and a new virus particle could be produced. So this are the certain interactions that takes place, and then all the virus infections that leads to triggering the production of interferons, right? So interferons already have discussed in details, where you know that these interferons produced by the virus infected cells, and they try to protect the other adjacent cells for further virus infection. So this is another phenomenon that happened in the due to the virus infection to the host cell. Now, coming to this persistent infection, so the literally meaning is that the virus is not eliminated from the cell. There are many infections where the virus enter into the body, then due to the effective immune response, they will able to eliminate entire virus population from the body, and the animal will be recovering from the disease. So complete viral clearance will occur from the host. It's happened with many virus infections. However, in case of certain virus infections, maybe one population, not the entire animal, all the number, all the animals in the population, maybe a certain percentage of the animal, the virus may undergo persistent infection. That means the virus is not eliminated from the cell. So usually we call them as a chronic type of infection. There is no or limited clinical manifestations the animal will show, but it will harbor the virus for a quite virus persist inside the host for a persistent infection. The viruses are replicating at a very slower pace, and only few number of virus particles are inside the host cells, or maybe because of several reasons. So one reason is the host immunity highest state, then the virus will not get the chance to grow and multiply and comes out of the host cell. So in the chronic stages, if the virus grow and produces the virus particle and slowly release from the in less number through certain secretion and excretions, we call them as the persistent productive infection. And generally it is designated as the chronic type of infection, right? However, the word latent infections, where after the acute phase of virus infection, the virus will not be produced. Practically, there will be no replication of virus takes place, but it will remain within a cell at a specific location without producing any infection. We call it the persistent productive infection. These are some very critical things that we could see in certain virus infection, more particularly the herpes virus infections. After the acute phase, the virus undergo latency, particularly in the trigeminal and sciatic nerve. The virus remain in a latent state. No productive virus infection takes place. This viruses may undergo acute phase of the disease. So that kind of stage is called as the latent virus infection, and it is extremely difficult to eliminate and to detect those animals where the latent infection is present. Okay? Again, there are certain phenomena where in certain virus infection, if the virus gets exposed to the transplacental transmission, if it occur to the newborn babies. So during their maternal period or during the pregnancy period, if the virus infection enter into the fetuses, then those, not in all, but in certain cases, certain diseases like the bovine viral diarrhea virus infection, the newborn cough will show no immune response against that virus. However, a large amount of viruses will be produced into that animal, and they will become a source of infections. And the animal will not show a single symptoms of the disease. However, at the later period, suddenly when they come in contact with certain wild counterpart or wild type virus, then they things that we discuss when we talk about certain specific virus infections, more particularly to this bovine viral diarrhea virus. Again, we'll mention about that aspect. So second means no active virus particle produced. We call it the persistent non-productive infection, right? And the third category, the last category, is called as the transformation or the cellular transformation. So it may be the acute or it's an outcome of persistent infection. Many of the virus infection. So to understand this viral persistence, why it is important for us, the persistent viral infection, it's because the apparently healthy animal doesn't showing any signs of the disease. All of a sudden, the reactivation may occur from the what we call, and an explosive disease may occur. So without getting any clue of the presence of the virus, suddenly the virus infection may take place. And this reactivations may causes a severe outbreak of the disease. Generally, in case of the human herpes, nobody knows when the reactivations will occur, and all of a sudden, so you may develop the painful blister formation in the skins and the nerve tracts, and the reactivation of the virus may take place. So this is why this persistent infection. So most of the virus infections where the latency occur, always it is suggested, in case of the animal, always it is suggested to eliminate those animals who got exposed to the disease, and so that they cannot be a source of infections in the herd. Then in the persistent infections, when in the chronic stages, persistently when the virus produce, and there's very slower rate, and the replication of virus will take place in a very slower space for a prolonged period, that may lead to some immunopathologic condition. Immunopathologic condition, that means there are certain cases where the persistent presence of the virus in the kidney tubules may lead to activation of complement system. There are certain conditions where the anemia occur. Presence of the virus at the chronic stage may lead to hemolysis. So some of the immunopathological conditions are produced because of persistent infection. Persistent infection is also responsible for neoplasia or tumorigenesis or transformation of cellular transformation. So persistent infection is again important because in the vaccinated animals in a herd, the animals may carry the viruses in persistent form. So the last one is a source of contagions or in the long distance viral transports and reintroduction with this virus infection. The persistently infected animal, when it is apparently the animals are healthy, but they harbor this agent, and they become the source of infections or introductions of new diseases may take place in an area. So this way, these persistent infections are important. Note down the different types of infection, slow infections, or acute infections with late clinical manifestation. These are different types of persistent infection. So persistent infection is the interaction between the virus and the cell. Okay. Through certain diagram, just as we see this one, what basically happened in acute infection is that once the virus will enter and the animal will start showing the clinical manifestations and goes to the highest level, again, virus particle will be remain. This is what we call as the acute infection. But in this graph, if you see that the acute infections occurring in an animal, but the virus particles, after a definite decline, they remain stable and they remain inside the host for a long period, what we call as the persistent infection. So this is the graph that we can draw it to explain the persistent infection. So the virus continued the replications speed or pace, but it remained for a longer period. Then in this latent infections, what happened after an acute phase of disease, the virus is no active virus particle will be formed. It will come to the baseline like this way, but the virus will remain within certain cells. No active virus particle are produced. That's why it is the graph, you can see it at the base level. And due to the reactivation, again, the recurrent infections and the episodes of the disease will again at different stages. Like this way, an animal throughout its life may show this kind of reactivations and disease episode. So when this disease will occur, this is the active stage where they can transmit the infection to other animal into the population. So this some of the pictorial or graphical representation of acute, persistent, latent infection. This different condition. Yeah. Here, coming to this particular, it's an example of the rotavirus diarrhea showing the acute disease. The disease clinical manifestation occur. Then this is the phase where the mortality or the death occur, episodes of the illness, and again the clinical manifestations are that the virus is the decline, and finally. So we call it an acute kind of disease. But when we say it's a latent, that means the disease episode will totally, no more virus particle will be produced, but the virus will remain in a static state. Reactivations occur at the different phases. Whereas even some reactivation, no clinical manifestation may also occur. Then this is caused the chronic infections, the congenital chronic infections, we say. So the virus will be producing without any clinical manifestations, and finally the chronic stage of the disease may lead to the death of the animal. So this is a typical, again, chronic infection. In the foot and mouth disease virus, so clinical manifestation will be shown, the acute phase of the disease, and gradually the virus will decline and they will persist for a longer period. So this is a type of chronic persistent infection. And these particular stages, they may act as a reservoir or can source of infection for an animal. Then some of the like the acute, occasionally chronic condition in canine distemper, you can see after the acute phase, the virus will remain for a chronic period. Sudden death may occur because of encephalitis or neuronal loss of neuronal functions. And to the late phase of the disease, the terminal nephritis or encephalitis, old dog encephalitis may occur and the animal may die. There is a type of, again, persistent infections called as a slow virus infection. We're going to study some slow virus infections like your scrappy infections, and we are going to discuss about the Visna and the Medi retroviral infections, where it took a long time, even 1 or 2 years incubation period, and gradually the virus particle will increase inside the host, and finally it leads to death of the animal. So these are some of the different types of persistent infection. So in this topic of the virus cell interactions, at the end, how we can summarize this whole aspect is that there are basically 4 types of virus cell interactions. So first is called as the cytocidal interaction. In the cytocidal interaction, the growth and the presence of the virus inside the host cells will lead to cytolysis. The cell damage will occur. There are different mechanisms that we have discussed, including the immunological activation or immunological mechanism of cell death. It may occur, and the active virus particles are produced and come out, and they infect the other adjacent cells. So the damages may occur at a different level: the level of nucleic acid synthesis, the level of protein synthesis, messenger RNA processing, then the plasma membranes, fusion, syncytia formation, heme agglutination, heme nitrogen, the different mechanisms through which these cellular changes, the architectural changes may take place. The second type of viral cell interaction is the persistent infection. In the persistent infections, we have 2 different categories. One is the persistent productive infection, where the active virus particles are produced in chronic diseases, and they are slowly excreted from the host cells for a prolonged period. The other category is the persistent non-productive infections, which also we call it as a latency or latent. No active viruses are produced until and unless they are reactivated. So once an animal gets exposed to the virus, and a period of acute disease will occur in the animal, and after that the virus will hide inside certain specialized cell, or we can say it is remain in a dormant state. There are different mechanisms, and no active virus particles are produced for a period, and again the reactivations and new episode of the disease may produce. So that kind of virus cell interactions, we call it as a persistent, non-productive, or latent virus infection. So these words are very common while explaining the viral pathogenesis and epidemiology. So it used to be customized with these different points. Then the 4th type of viral cell interaction is the transformation. So transformation is basically referred to all the oncogenic viruses or tumorigenic viruses. So there are many viral infections where the cellular transformation occur. As already have studied the Marek disease in poultry, avian leukosis in poultry, where the T cell transformations occur that leads to development of solid tumors. So those are the tumorigenic virus infections. Do you have any queries or anything here in this chapter? If not, please do text me and put some questions so that we discuss in the subsequent class. And the next chapter on which we need to highlight is the genetic and non-genetic interactions among the viruses. It's quite interesting topic, the genetic and non-genetic interactions. This genetic interactions refer to like the virus genes products, how they interact with each other, the virus genes, genetic interaction. And this is one reason why we could see the emergence of new viral disease and infections more frequently than that of the bacteria and other diseases. So it has been seen that there are a certain category of viruses where they exchange their genetic material, and that exchange of genetic material, or shortly we can say the genetic interactions, may lead to emergence of a new type of viruses and with a new disease outbreak in the population. So at the same time, it is happened like there are certain virus interactions where it is non-genetic, means the genes doesn't play a role. Rather, the presence of two viruses in the environment, they sometimes help each other. There's a positive interactions, or sometimes presence of one virus prevent the second virus infection. So it's a negative type of interactions we could see among the viruses. So collectively in your syllabi, this topic has been given: genetic and non-genetic interaction of the viruses. So some aspect of it we'll try to discuss in this session. Okay. So the viral gene product, the genetic interaction of the viruses, or maybe the environmental interaction. The presence of the virus changes the environment, and that may favor or not favor the growth of the second virus. And thirdly, the immunologic interactions. So as we know that the immune response that produces against the virus infections protect the individual from the subsequent virus infection. But sometimes it's seen that some of the primary immune infection, these are very, very complex mechanisms we could see among certain viruses. More particularly dengue, you'll be surprised to know that the presence of some circulating antibodies, non-neutralizing antibodies against the dengue virus, favors the second infections. So the second infections may be more severe than the first one because of the presence of certain non-neutralizing antibodies. Likewise, we could see some sort of interactions among the virus. Okay. So the direct interaction of viral gene products, here we are going to give emphasis more on the genetic recombination process, genetic recombination. Of course, others are also occur among the viruses, and some things that mostly we discuss in the postgraduate level, but here we give more emphasis on the genetic recombination. Shortly, if I say the helper-dependent virus is that one virus protects and helps the other viruses for complete its replication. So probably in the previous class, I mentioned that the virus replication process is not a perfect process of replication. Many defective virus particles are produced. So a defective virus particle always need a proper virus to complete its replication cycle. So there are certain examples where we can say that the vaccines, what is prepared in the market against the SARS-2 coronavirus. So it is an RNA-associated virus vector, and which always needed an herpes virus to complete its replication cycle. Alone, this adenovirus, which what we say is a modified adenovirus, it cannot complete its replication unless the herpes viruses genes are present in the environment. So they helper dependent. One helper virus is needed to complete this replication cycle. So likewise, we could see many examples of it. So the meaning of helper-dependent virus is that among the two viruses, one virus helped the other virus to complete this replication. Then there is a phenomenon called as the phenotypic mixing. This is again a very unique things that we could see among the viruses, not seen in other organisms. So two related virus, unrelated virus. When two different related viruses, say virus A and virus B, with some colored diagram, it has been showing the genome blue, capsid blue, genome black, capsid black. So these two viruses, when they causes mixed infection, that means both the virus, when they infect a same host cell, it may be possible two viruses may infect the same host cell. The progeny virus, when they get assembled, then you may get as a virus A pure form, the virus B in the pure form, but these two categories you can see the capsids where the nucleic acid is of B, but the capsid is a mixture of A and B. This is the method what we call as a phenotypic mixing, usually seen among the viruses. So the other one where the genome is of A, but the capsid is a mixture of A and B. So likewise, this virus, further when they replicate, this property gets lost. Like whatever the genome is there, that genome capsid will produce in the next generation. So here also, but at this stage when they infect the same host cell, this kind of phenotypic mixing may occur. There is a condition what we call as the polyploidy. Suppose this particular virus, when they infect a host cell, instead of one copy of the gene, it may accommodate 2 or 3 different copies of the gene within the nucleic acids. We call it a polyploidy. Even a condition called as the heteroploidy. So this and this two may get accumulated within one capsid. So this kind of interactions we call it a phenotypic mixings, and it occurs because of 2 viruses when they infect the same host cell. Usually it's seen among the related virus. Now coming to another condition is called as the superinfection exclusion. You understand the word superinfections. We know that this word very frequently we use it superinfection in case of long-term take the antibiotics for prolonged period. So normal microbiota reduces, and that favors the growth of fungus, particularly the candida infections inside the body. Basically, what is happening about this black fungus these days is becoming a hot topic, and how many corona patients are developing mucormycosis, the black fungus infection. This black fungus, basically the mucors are very common saprophytes. You'll get it in your environment. Just keep a piece of bread, you can see the mucor growth is there. So mucors are floating in the air all the time, and we are not suffering from any infection because of the mucor. The way it is projected in the media and the news channel is not very for spores, and we are not suffering from any infection. It's basically like until and unless a person's immune system is compromised, the special cases of the SARS infection, where the immune system goes down, and then to reduce the inflammatory conditions when the steroids are given for a prolonged period, then we know that steroids are again the immunosuppressant drug. So the immunity goes down. In that case, if through certain medical equipment, particularly maybe in inhalers, if many mucors get inhaled inside the body, then they can set up infections and some hypersensitivity reactions may occur. But in normal healthy individual, it's not going to do anything, this mucormycosis. So the superinfections, like the presence of treatments, infections that way. Here the cases is that superinfection exclusions means they will the presence of one virus infection prevents or block the second virus infection. Different examples are there. I'll be highlighting only the equine infectious anemia virus infection. This is in equine infections. We'll be discussing subsequently. So this virus, when they grow and multiply inside a host cell, they secrete a soluble protein that will block its receptors so that the second virus infections doesn't occur. So this is a mechanisms for the survival inside the host and to become a privileged area inside. Now coming to this topic, this is important. Should I continue for another 15 minutes? Sure.
Yes, sir.
Can I just continue?
Yes, sir. Yes, sir.
Okay, thank you. The genetic recombination. In fact, I will tell you, no doubt that this present pandemic, there was a time, almost 10 years back, the WHO, there is a speculation that this particular phenomenon, this corona has come up and nobody has speculated earlier about the corona, but still there is a high risk that maybe in near future we may experience a massive pandemic caused by the influenza viruses, and that is basically because of the genetic recombination process. This is a genetic interaction among viruses, more particularly the segmented RNA viruses, although it has been seen in other RNA viruses also, but very common, this genetic recombination is very common among the segmented RNA viruses. Okay, we talk about viruses, orthomyxoviruses, then Bunia viruses. These are examples of the segmented RNA viruses. So the influenza viruses or the orthomyxoviruses, they frequently undergo this genetic recombination. Of a segment of RNA, segmented RNA change takes place. Viruses. So to occur this particular phenomenon, the condition is that two influenza viruses, let us take the example of L and M, these are two different influenza viruses having the segmented genome. When they infect the same cell, same host cell, they must have to infect the same cell, the epithelial cells or anywhere, when they infect the same cell, in the process of assembly, when the new virus particle form, two different influenza viruses infect the same host cell, and when the progeny viruses will produce, we generally see a third type of virus. Of course, the original L and M will form, no doubt, but there is a probability that a third recombinant virus may produce, where they will carry the material of both the virus. If you can see here, it is depicted with the colored segments, the red color, blue color segments, L and M. And these are the red and blue color synthesis that takes place. And when they get assembled, here it is all the red segments, here all the blue segments. But in this virus, you can see they one segment. This kind of phenomenon are called as genetic recombination. Okay, so genetic recombinations give rise to new type of virus. Particularly, the surface proteins may get affected very frequently, and this particular virus will no more be protective against the antibody against these two viruses. Suppose these are the two common flu virus that we are harboring and we are having immunity against these two viruses. The recombinant virus may not be protected by that antibody. And this is a continuous phenomenon that we could see among the influenza viruses throughout generation. And sometimes some of these recombinant influenza viruses leads to a massive pandemic that we could see in the past history, like the Spanish flu that's responsible for death of more than 50 million people throughout the globe. Spanish flu that lasted from 1915 to 1970. Within that 3 year, 50 million people they died throughout the globe. So the very recent time in '65, the Hong Kong flu, and then they also kills around 3 to 4 million people throughout the globe. So the pandemics has really appeared very badly into the human race, and all of a sudden this virus is protein or recombinant virus may produce naturally occur. Certain more aspect about this genetic interactions and recombination, how it may happen. Like it says that the migratory fowls, the water birds, they are mainly harboring from distant places. They can come and here. So when this bird flu virus, when they come in contact with this within the human host, then this two virus recombination may occur. Okay, so this is more particularly called as the reassortment. Sorted or reshuffled, we can say, and that may give rise to development of a new virus, which may have the characteristics of this as well as this. However, this particular virus may be a common flu virus, not affecting very severely in the human. But when this virus gets recombinant with a wild type virus comes from the migratory water bird, may lead to, not all the times, may lead to a new strain causes influenza outbreak. So likewise, the duck and the pigs, pigs are considered to be a mixing vessels, has been seen in many influenza outbreak where pigs are as a pet animals or as a livestock are bring by the human. They may get contact with the wild type virus, and in the pig, these two viruses may get mixed up, and that leads to adaptation of the new recombinant virus in the human, and human flu or influenza outbreak may occur. So usually these things are happening in the nature, and these viruses, they are exchanging or reassorting their genome segment, and that leads to production of new viruses, which may breach the immune systems and leads to severe outbreak of flu. Okay, so this phenomenon is called the genetic recombination. So as I said, that we'll be giving more focus on the genetic recombination. So because of this genetic recombination, a particular phenomenon that occur is called as antigenic shifting. So this particular virus is having a definite antigenic structure on its cell surface. And this structure, in case of the flu virus, it is spike proteins, H and N. These are the two antigenic structure. So when they combine with a different type of virus and new reassortment occur, the antigenic structure may get changed. The different H and N number may produce. So when it happens, the normal immunity against the flu viruses that the human are having may not able to restrict this particular virus for replication. So this particular phenomenon is called as the antigenic shifting. The antigenic structure may get shifted because of reassortment. So this is a very important genetic interaction, but not only the influenza viruses, the similar kind of exchanges also we can see among the Rio viruses, where we can see several serotypes of blue tongue virus infection in goat. We are going to discuss subsequent class. So this particular phenomenon is called the reassortment. Okay. Then there is another again, it's called an embedded virus. Embedded virus is not very common, it's uncommon, but it has been seen in the fowlpox genome. The reticuloendotheliosis virus, entire genome of the reticuloendotheliosis may remain integrated with the fowlpox, the genomic structures. And when this fowlpox carrying this reticuloendotheliosis virus genome, when they affect the poultry, they may cause a severe immunosuppressive disease in poultry, and high mortality may occur. This kind of example is called as the embedded virus. Of course, the examples are very limited. Another phenomenon is called as the heterologous transactivation. So let's get this one. We'll not much discuss about it. one. So the direct interaction of viral gene products mainly refer to this phenotypic mixing and pseudoviruses, then transactivations, then we talk about the genetic reassortment. These are the phenomena. So in the next class, we'll talk about the indirect environmental interaction and immunological interaction. Okay. Thank you very much for attending the class. I guess you should have a lot of questions on this aspect. Please do read the books on the veterinary virology by Fenner or Murphy. Both are equally good. Or even you have the Queen's microbiology, that also the virology sections you can study. If further explanation or some more things are needed, you can discuss and put some questions. We can have some question session in one class and can interact. Thank you very much.
Sir.
Yes.
Sir.
Yes.
Sir, what is the difference between phenotypic mixing and genetic recombination? Both are.
Yeah. Genetic recombination is a mixture of genes, genomes. But phenotypic is the capsid protein, mixing of capsid protein. Could you see? Only the capsid protein get mixed, not the genes. So phenotypic mixing means the phenotypic characteristics. You understand the phenotype. Phenotype means the appearance. Genotype means the genetic constituent. So reassortments leads to development of a new virus, but it doesn't call it as a phenotypic mixing. Yes.
Okay, sir. And sir, what is pseudotype viruses?
A pseudotype virus means it's look like the virus, but this is an incomplete virus. So they may have a capsids intact, but the genome is less in number during the assembly. We call them as the defective virus. Pseudotype virus are look like the original virus, but they don't have the capacity to yeah, there are many informations are there, but considering the undergraduate courses, so we need to restrict at certain points. Of course, this is quite interesting. We can discuss, no issue. You can text me the questions so that I can address in the next class, as previously somebody with the answer that I have given at the beginning of today's session. So one thing is very clear about this, why the protein synthesis takes place in the cytoplasm, because it always occurs in the cytoplasm, not in the nucleus. But assembly may occur in the nucleus in case of certain DNA viruses. So in general, the DNA virus, they replicate in the nucleus, and the RNA virus, they replicate in the cytoplasm, with certain exceptions. So exception you must remember. So in case of the RNA, the exception is the influenza virus, and in the DNA, the exception is the African swine fever virus and the pox virus independently.
Thank you
if
very
no more
much.
questions, then you can please text me any queries if anything is there.